Good evening, everyone. I welcome and again to our classes for Tailored Soft Academy. Today, we're going to discuss an interesting topic, and what we discuss now prokaryotic and eukaryotic cells. Now, what this thing mean? We find out, say, the time when science been begin, the first major challenge we many scientists face that time. Now, which group to put which organism for? The world been get so many different organisms. These organisms they go to thousands and millions, and it can be very difficult to put these organisms for different categories. Now scientists they ruminate this matter, they can't come say okay, they can't decide say maybe they hit on a particular method, say okay, they go divide cells into two major classes. Now within these two major classes of cells, now either prokaryotic cell or eukaryotic cell. So we'll go look at different examples. Now prokaryotic cell, which example they? Prokaryotic cell go be either bacteria or it go be archaea. And if na eukaryotic cell, it go be either plant cell, it go be animal cell, it go be protista, or it go be fungi. Now the question come be, okay, these prokaryotic cells, wetin them be like, wetin their structure be like. We'll take some time to look into the structure of a prokaryotic cell, wetin dey there. Now a prokaryotic cell, the first thing wey a prokaryotic cell get na say e no get nucleus wey dey as defined as a eukaryotic cell. For prokaryotic cell, e get wetin we dey call nucleoid region. And wetin that mean na say all the genetic materials, all the DNA, all the things wey dey move traits from generation to generation, e dey together for one area of the cell. And that area of the cell, you no get any membrane wey cover that area of the cell. So we dey call that area of the cell the nucleoid region. And na wetin dey for prokaryotes be that. Prokaryotes get nucleoid region. Prokaryotes get cell wall, and now wetin we dey see for bacteria be that. Prokaryotes dey get cell wall. Now if we look at the size of prokaryotes now basically between 0.5 micrometer to 5.0 micrometer. Now wetin be micrometer? Micrometer na the measurement wey we dey use measure bacteria. And how we go get an idea of wetin be the size of this micrometer? The way to look at am na say if you take ruler, and for that ruler you look at 1 meter for the ruler. If you fit divide that 1 meter into 1 million places, each of those places now 1 micrometer. So imagine now the size of bacteria now between 0.5 to 5.0 micrometer. That is the size. If you want to understand that more, think about the human hair of a human being. The thickness as it take thick now 70 micrometer. So the size of bacteria now between 0.5 to 5.0 micrometer. Now apart from the size of the bacteria get cell wall, and then bacteria they involved. That bacteria na prokaryotes. So prokaryotes dey involved in many activity. This activity we dey call them metabolic activity. And wetin we mean? All the activities wey bacteria dey run in order for the bacteria to keep alive. Now these activities, sometimes we dey divide these activities into aerobic or anaerobic. And wetin that one mean? Any activity wey the bacteria dey go into wey be say dey make use of oxygen, that activity we dey call am aerobic activity. If e dey do that activity and e no dey use oxygen to do the activity, we dey call am anaerobic activity. The same bacteria dey also undergo fermentation. So e get many activities wey bacteria dey do, and all these activities e dey do am in order to be able to sustain life. Now if we take the other side wey be eukaryotes, eukaryotes get their own characteristics. The first thing wey we know about eukaryotes na say they dey at a higher level of specialization. That one means say they dey more specialized, they dey more developed than the prokaryotic cells. Sometimes we dey refer to the prokaryotic cells as primitive cells. Meanwhile, we dey look at the eukaryotic cells, we dey call them more specialized cells. Now wetin make them dey more specialized? The thing be say if you look inside the eukaryotic cell, you will notice say many features dey inside, many structures dey inside, and these structures we dey refer to them as organelles. And wetin organelle mean? Different types of organelles dey. We get ribosome, we get lysosome, we get mitochondria, we get nucleus. Now all these structures wey been dey inside the eukaryotic cell, they get membrane wey cover them. Now one very important feature wey eukaryotes get, wey prokaryotes no get, na say eukaryotes get membrane bound organelles and they get nucleus. They get nucleus wey dey well defined. Now wetin be nucleus? Nucleus na that area of the cell where all the genetic materials dey for inside that area. And wetin that one mean be say that area na the area where they control the activities of the cell. Anything wey the cell wan do, e go get instruction from the nucleus of the cell. So the nucleus dey and the nucleus dey inside the eukaryote. Now some eukaryotes dey get cell wall if say na plant cell, but if e no be plant cell, if na animal cell, e no go get cell wall. And examples of eukaryotes, like I said before, fungi na example of eukaryotic cell. Protista like amoeba na another example. Then any plant cell, whether na flower, na leaf, or na tree, any plant cell na eukaryotic cell, and also any animal cell go be eukaryotic cell. Now we go complete this class by trying to look at wetin be the differences wey dey inside prokaryotic and eukaryotic cells. They get major differences. Number one, the prokaryotic cell no dey get defined nucleus. E no get a particular defined nucleus. E no get any membrane wey surround one area wey we dey call the area nucleus. No, rather e get wetin we dey refer to as nucleoid region, wey be a region or an area in the cell wey be say genetic materials boku for that area. Meanwhile, the eukaryotes dey get correct nucleus wey get membrane all around it. Now the prokaryotes no dey get organelle. They get structures inside them wey dey help them run many metabolic activities, but they no get organelle. And the meaning of organelle na that structure. Once that structure get a membrane around it, we refer to it as organelle. So if you get a cell now, the cell wall go dey or the cell membrane go dey, then structures go dey inside the cell. For eukaryotic cell, those structures go get small membranes around them. So we go dey call them organelles, whether na mitochondria, whatever organelle e be, we go dey call them all those names. But for your prokaryotic cell, e no get such arrangement. You go just get your nucleoid region where things plenty for, where genetic materials plenty for that area, and then other things go dey inside the cell where they help the cell with other activities. Now for genetic materials, we notice say the prokaryotic cell, that one will be your bacteria, e get wetin we dey call single circular chromosome. And wetin e mean be say the genetic material na like a single one wey dey circular, but they come coil am plenty. Na very large thing wey dey coil plenty. E come sit down for one region of the cell, and that region na the region wey you keep to dey refer to as the nucleoid region. Meanwhile, for your eukaryotic cell, they get multiple linear chromosomes. And wetin that one mean na say many chromosomes go dey straight in lines plenty. Now if you look at human cell gets 23 pairs of that kind of thing wey we dey call chromosome. And that chromosome, e dey carry genetic materials, e dey carry information for the cells, and that na wetin the eukaryotic cell get. Lastly, when it comes to the issue of reproduction, we find out say the prokaryotic cell, the major method wey dey use do im own reproduction na wetin we dey call binary fission. And binary fission, e dey easy. Wetin dey happen for there na say the first cell go dey wey we dey call mother cell, and if you don grow reach a particular point, e go divide, come form two cells wey we dey call daughter cells. So single mother cell go come form two daughter cells, and this na wetin we dey refer to as binary fission. And that is the major way or the major method wey our prokaryotes dey use take reproduce. Meanwhile, our eukaryotes no be so. They get many methods. They use wetin we dey call sexual reproduction. They also dey use asexual reproduction. So many of these things we go dey discuss as we go along in the class. But the major thing for us to know na say for cells, two major types of cells dey. The cell go either be prokaryotic cell or e go be eukaryotic cell. If na prokaryotic cell, na cell wey never develop very well. We dey look am as a primitive cell. Meanwhile, if na eukaryotic cell, na cell wey don dey develop, wey dey more specialized. Thank you for listening to this class. I been hope say I try to make sense with these few things wey I talk. Have a wonderful day, everyone.